There was an article I was reading, I'm trying to think where it came up. My wife's boss is a chemical engineer, and when she reads some of these scientific magazines, she sends it along to me, and there's one about shortage of organs and different hopes for the future. And I'm reading this article, as I always do, and there's this particular paragraph that's talking about a David Cooper who is doing research, and I'm trying to, I won't have the detail, I'll let him explain that, but I'm going to try anyway. He started out someplace. He ended up in Australia, if I recall correctly. Am I right or wrong?
South Africa.
South Africa, close enough. Not where he started or where he ended up. And the thing that caught my eye was he's now at the University of Pittsburgh, or Pittsburgh Medical Center, wherever. And I thought, well, we're having this conference. This is a great topic for us. I wonder if this eminent person would mind come talk to us. So I did some research, found his email, sent it to him, and within an hour, got an email back saying I'd love to share what's going on. He's got a fascinating topic that's talking about xenotransplantation. It's losing some of its high profile right now. I thought what a great way for him to bring his message to us and put it right on the forefront for us. So David, please come forward and talk to us.
Well, thank you very much.
Yes,
yeah. I think you can all hear me, yeah?
Yep.
Yeah.
Yes.
I'm going to give you a little summary of what's going on in this field of research, which is xenotransplantation, which actually means cross-species transplantation. And obviously relevant to you and me is the transplantation of using an animal organ or cells in patients, in humans. So it's cross-species in that respect. And we're trying very hard to be able to use pig organs for a number of reasons. They're very similar in structure and function to human organs in many ways. And pig cells, such as islets for diabetic patients and so on, or corneas for corneal transplants. Because this would obviously solve the problem of donor supply, and which is the biggest problem around really at the minute. I used to be a heart transplant surgeon. More patients died waiting for a transplant than they died after the transplant, obviously. So this is the biggest problem at the moment. So I'm going to run through it fairly briefly, and it's obviously what I'm going to present to you is not my work. It's a very large group of us at the University of Pittsburgh, including colleagues at the Children's Hospital as well as in the Starzl Transplant Institute, and colleagues down at a small biotech company called Revivicor, which is in Virginia. And this company, which has about 20 staff, concentrates its efforts on genetic engineering of pigs for medical purposes, particularly for this idea of using them as organ donors and so on. But also they have another approach, which I won't go into, where they try to make human immunoglobulin, human plasma, in pigs. We make our own immunoglobulins, our own antibodies and so on, and you have to pool antibodies from large numbers of patients in order to treat various conditions, whereas if they could make human immunoglobulin in pigs, and they can do this actually, then you could use pigs as the source of all this human immunoglobulin. You wouldn't have to bleed patients, you wouldn't have to worry whether the patient's got HIV or hepatitis or what it is. So it would be another big advance. And this company arises, and some of you may remember in the 1990s, there was the Dolly the sheep phenomenon, where Dolly the sheep was the first cloned mammal. They took a cell, an adult cell from another sheep, and cloned a new pig from that one cell. And that's what this company is, an offshoot of that, and this is what it does. But now it can actually take the cell, it can do some genetic manipulation in the cell, in the laboratory, and then clone the pig from that cell, including the genetic manipulations. And it can actually knock out a pig gene, if we want to get rid of it, or it can put in a human gene. And we now have pigs with 5 different manipulations. 1 pig gene knocked out, 4 human genes put in. I'll come back to whether this makes a pig partly a human or not later on. But, and in some of the cases, we may well need at least 10 different genetic manipulations before the pig is ideal as a donor for humans. And here's the sort of technology they use. They use powerful microscopes, and they put the cell under the microscope, and you can see just to the right of the picture that there is a very, very fine pipette here, a very fine needle, and they can put in or take out some of the genetic material. And they're becoming very skilled at it. Now, I like this quote. I'm sometimes reminded of the differences between a rhinoceros and a cardiac surgeon. One has a thick skin, a small brain, and charges a lot. The other is a large animal, lives in Africa. Now, this was said by one of my colleagues in Britain, where I come from, and you're only allowed to make this joke if you are a cardiac surgeon, otherwise we consider it very insulting. But there's an earlier quote, 1st century, there are 3 ways to treat a patient: diet, medicine, and surgery. But only surgery works, and that's very true even to this day. Now here's some information that you will find interesting. If you take chronic kidney disease, CKD here, chronic kidney disease in the United States, you can see that we spend each year over $50 billion on the treatment of patients with chronic kidney disease. Of that, about 20 billion goes on dialysis and so on, and a very small amount on kidney transplantation. And kidney transplantation is much cheaper, believe it or not, than chronic dialysis. And so the problem we have so many patients with chronic kidney failure and still on dialysis is largely because we don't have enough donors to deal with them all. So it would be a big economic boon if we could transplant patients instead of keeping them on dialysis. And remember, hang on a minute, remember that if you came in with kidney failure, if you had an unlimited number of donors, you wouldn't have to go onto dialysis. You'd have the kidney transplant the next day. And that would save you years of chronic dialysis, going to the dialysis clinic every 2 or 3 times a week and so on. So it would be a huge step forward. Did you have a question?
I just had a smart remark.
Go ahead.
Did you send that to our U.S. legislators when they're deciding whether to pass the bill to continue to fund the kidney post-transplant medication?
I'm sure it comes from the legislators or the legislative staff, but they probably don't, look at it. But it is quite true. And almost a third of the entire Medicare budget goes on chronic kidney disease. It's an enormous amount of money.
Wow.
Now, this is the problem that we're faced today, as you all know. The red line is the number of patients on the UNOS waiting list in the United States up to 2011. It's about the same this year. About 110,000 people waiting for a donor of one sort or another, heart or kidney or whatever it is. And the blue line is the number of donor organs that become available from deceased donors each year. And you see it's around about 30,000. It's not the number of donors. The number of donors is actually about 7,000 or 8,000, but we get at least 3 donor organs or 4 out of each donor. So we've got about 30,000 organs to treat 110,000 patients. So you can see that a lot of patients will have to wait till next year, or they will die while they're waiting. Maybe a assist device will help those who are waiting for a heart. Dialysis help those with a kidney. Those waiting for a liver may not make it, and so on and so on. And so it's a major, major problem. Now, if you want to read about this in more detail, I wrote a book about 10 years ago called Xeno here, which covers this whole topic of can we use animal organs and cells for the purposes of transplantation in patients in simple terms. And it's quite interesting. It's not only from a scientific point of view, but from a legal perspective, an ethical perspective, and so on. You see that the foreword we got was written by Robin Cook, who some of you may know is a well-known novelist. He is a doctor. He was an eye surgeon, actually. And he very kindly wrote this. And in Amazon.com, this book was entitled Xeno by Cooper, Lanza, and Cook. And I thought I'm going to sell thousands, millions of copies because people will think it's Robin Cook's next novel. But unfortunately, people got wise to the idea. But it was good of him to write. But he has written books like Coma and so on, which have done extremely well. So if you really wanted to learn anything about, read a bit more in detail about this book, I think would tell you it's still available in places. Now this is an Englishman called Sir Peter Medawar, who is considered the father of transplantation immunology. He wasn't a surgeon, he was an immunologist, a laboratory guy. And he did the early work in the late 40s and early 50s showing that you could transplant and you could actually get tolerance to the transplanted organ if you manipulated the recipient in some ways or other. So he won a Nobel Prize for this in the late 50s, 1960. And he said at that time, 1969, he said, we shall solve the problem of organ transplantation by using heterografts, which was the old word for xenograft, meaning using animal organs, one day if we try hard enough, and maybe in less than 15 years. So you can see even Nobel Prize winners get it horribly wrong, because here we are, 50 years later, still trying to solve the problems that he thought we might be able to solve in 15 years. So it's fooled most of us for most of the time. And this is a good example of this saying that predictions are risky, especially about the future. Now, I don't know many predictions that are not about the future, so I think that gives you a good idea of the standard of some of our politicians, or it may be Yogi Berra, the New York baseball guy. Now, the idea of xenotransplantation goes back a long way. Here's a mythological figure. This is a lamassu, but there are lots of others like mermaids, chimeras, and so on. You can see it's a mixture of different animals, bull, human, bird, etc., etc. And so the idea of mixing animals is not a new one. And as early as the 17th century, 18th, 19th centuries, quite a few people were given blood transfusions from animals. Here's an example of a donkey giving the blood, going into a patient. It was thought it was good treatment for mental disease. People who were mentally deranged would be given blood from sheep, because sheep were thought to be nice and docile animals, and it would calm the patient down. I'm sure it did calm the patient down, but it would kill a few of them as well, I think, because it's not without a lot of hazard. But it went on until the early 20th century, believe it or not. And then, in the early 20th century, this guy on the left, Alexis Carrel, was a French surgeon working in New York, who started to do transplants. And he designed a technique for stitching two blood vessels together. And obviously, if you can't stitch blood vessels together, you can't transplant an organ. So he won a Nobel Prize for that in the early 1900s. And here he is with the guy that first, Lindbergh, the guy that first flew solo across the Atlantic on the right of the picture, because Lindbergh went to him and said, I've got this sister-in-law who has heart valve disease. Why can't we give her a new valve? And they didn't have a heart-lung machine at the time. And so they began to work on some simple devices to try to store hearts and so on, with a view eventually to try and develop a heart-lung machine, which didn't come about at that time until the 1950s. But Carrel, who was very interested in transplantation, in 1907 he said the ideal method would be to transplant in man organs of animals easy to secure and operate, such as hogs, for instance. But it would in all probability be necessary to immunize organs of the hog against the human serum, which is exactly what we're trying to do now. We're trying to protect the pig, manipulate the pig in such a way that its organs will not be damaged by the human blood and the human cells in the blood that will cause injury to it. So we're trying to immunize the pig in a way against the human serum. And he says the future of transplantation of organs for therapeutic purposes depends on the feasibility of xenotransplantation. That was his thing, more than 100 years ago. So he got it right, and 100 years later we're still trying to do what he predicted we would need to do to solve this problem. Now, in this brief summary of the development of xenotransplantation, we now come to the 1920s. And this guy, who was a Russian working in Paris, and he had the idea that if you have an endocrine deficiency, such as you've got no insulin, that if you put in the cells that make the insulin, then you'll solve the problem. And you can do this with thyroid disease or whatever other disease you've got, which was great visionary for the 1970s. His name was Voronov. And, which is just what we're trying to do now. We're trying to cure diabetes by putting in human cells, the insulin-producing cells, but we in our lab are trying to do this by putting in pig cells, which make insulin. I'll come back to this later. But Voronov was a bit ahead of his time, because one couldn't do it then, but you can do it now to some extent. but he was mainly interested in treating older men who were running out of their sex hormones, androgens and this sort of stuff. And he had a lot of men who were getting very weak and disabled, who the spark was running out of their lives, if you know what I mean. And so he started treating them with chimpanzee testicles. And he would chop them up and into slices and put them into the human's testicles. I don't think too many people came back for a retransplant. And he claimed, and this caught on, not only in France, it caught on in Britain and the United States, and several hundred or thousands of these patients were undergone. There was a guy in the States who actually used goats because he thought goats were very sexually active and so on. And they wrote reports that these patients did extremely well. I'm sure that none of them did any good except they gave them a lot of pain in the wrong place. But he was really working towards the right idea, but obviously he wasn't doing the right way. Now, you see here, in this picture here, he looks to me as if he needs a little bit of chimpanzee kidney himself to give him a little bit of a lift. He looks a bit jaded there, but this is quite true. At this stage of his life, he had been married to a Texas oil heiress, and she had just died, and she left his institute in Paris, a lot of money, and I have a friend there at the minute whose research is still funded by the money she left to this institute.
Wow.
But just after she died, he married a 21-year-old French girl, and he was by this time in his 60s. And I think that's why he's looking a bit jaded, not I make fun of him, but in fact he was a man ahead of his time. Now this is Keith Rinsma, who I knew well and sadly died a few years ago. And he was a professor of surgery at Columbia Presbyterian Hospital in New York. But in his younger days, he had carried out some transplants, some kidney transplants between chimpanzees and humans. And this was at a time when it was very difficult, even more difficult than today, to get a deceased human donor. And there was no dialysis, chronic dialysis. So he felt he had no choice, and so he tried about 13 of these transplants. And what he did, he transplanted both the kidneys from the chimpanzee, because the chimpanzees are rather smaller than humans, and put both kidneys in. And here is one patient, obviously at autopsy afterwards, and these are the chimpanzee kidneys, which actually look completely normal. There's no sign of rejection. And here are her own kidneys, her own native kidneys, which are terribly diseased. That's why she needed the transplant. She was the only one that did really well. The others survived about 6 or 8 weeks, and either died of infection because they gave too much immunosuppression, or rejection because they couldn't control the rejection. But she lived 9 months, and she went back to work as a school teacher, and she suddenly dropped dead, probably through some electrolyte, so potassium or magnesium or something like that. But I say her kidneys were in very good shape. And this was at a time in 1963 when the immunosuppressive drugs that we had were very, very primitive. And it was even difficult to prevent rejection and so on when you had a human kidney transplant. So it's quite remarkable. So with the drugs we have today, if we were going to use chimpanzees as donors, we'd probably do very well. They'd probably go several months or years even. But we're obviously not going to use chimpanzees because they're endangered species anyway. But this was interesting. At about this time, James Hardy, who was in Mississippi, he carried out a heart transplant from a chimpanzee. It was the first heart transplant ever done, actually, into a patient. He also died not very long ago. Delightful man. And what is interesting about that patient is that the heart wasn't big enough to support the circulation. The patient died after about 2 hours on the operating table. But this is the operative permit, the consent form that he asked the patient to sign. In fact, he didn't ask the patient because the patient was semi-conscious before this transplant. He asked the patient's family. And you can see, you can't read it, but it's only 1 paragraph long. And it says that no heart transplant has ever been done before, but it makes no mention whatsoever that they might use a chimpanzee as the donor. Can you imagine the trouble we get in today if we did this? We showed up and got the family to sign a consent form without mentioning we were going to put a pig heart in or something. But that was the norm for the time, and he didn't get particularly criticised for this because that was the norm for consent forms in those days. And then at about the same time, about 20 years later, Len Bailey, some of you may remember this, the baby Faye case. He had a baby who was dying, and he put a baboon heart in, and the patient, the baby, lived about 20 days and then died of rejection. Interestingly enough, he got a huge amount of publicity. I'm sure those of you who were around will remember this. Got a huge amount of publicity, and he told me that afterwards he got threatening letters for various reasons, and he had to wear a bulletproof vest for 6 months because he was being threatened all the time. But the interesting thing, I think, is that he got something like 700 or 800 letters complaining that he was experimenting on a baby. And he got about 17,000 letters complaining he was experimenting on a baboon, which gives you an idea of people's priorities. And here is baby Faye after the transplant. It didn't do much for xenotransplantation, but it did bring to the public's attention the difficulty of getting a donor for a baby in those days, 1983. And it started the whole idea of transplanting hearts in babies. And Len Bailey's results were superb after that. They were excellent. And babies rather do better than humans because their immune system is rather less developed and it's easier to manipulate it. And then, just after Keith Rinsma did his chimpanzee kidney transplants, Dr. Starzl here, when he was in Denver at the time actually, did some baboon kidney transplants, and later on did some liver transplant, including some in Pittsburgh where he used baboon livers in 2 patients, one of whom lived about 3 months. So there have been a number of attempts over the years. Now, if you look in the literature properly, if you know which medical library to go to, you find this going on much more commonly than you think. A woman receives pig's heart, now they can't keep her out of the mud. Man, 74, lives 19 years with dolphin's heart. Dying ape gets a human heart in history-making transplant. And I was surprised to read that this was done in Cape Town when I was working with Professor Chris Barnard down in Cape Town. And I don't remember this case at all, but evidently that was being done while I was there. And then Rich Ben's dog gets a human heart. So these transplants are going on all the time. You just have to know where to look them up. Now, the advantages, if we could use, say, a pig as the donor, are obvious. We have an unlimited supply of donor organs. We kill 100 million pigs in this country each year for food. So people don't mind killing pigs, and it would be a more honourable thing to kill the pig to be life-saving for some patient. So we have a limited number of donors of all sorts of organs. We would have the organs available electively when we want them. As I mentioned, you could come in today with heart failure and you could be assessed and they say, well, you need a heart transplant. Fine, we'll do it on the operating list tomorrow morning, 8 o'clock, because we can get a donor. It avoids the effects of brain death. All deceased human donors, as you know, virtually all of them are brain dead. And when you undergo brain death, it affects your whole body. I did a lot of work on this when I was in Cape Town. Your whole body stops using oxygen, believe it or not. You start to use up all your energy stores in your heart and so on. And that's why within 2 or 3 days your heart will stop beating unless you've given lots and lots of treatment. So brain death leads to the failure of all the other organs. And that's damaging to the organs. So that's why every now and then you put in a pig, you put in a human heart from a brain dead patient and it looks okay, but it doesn't work because there's no energy stores in it. Whereas if we use the pig, you would take the pig to the operating room, anesthetize it, and take the organs out under perfect conditions without all this injury going on about brain death. So it would be a big advantage and a big advance over what we do now. And you've probably heard quite a bit about, well, we don't want to give humans pig infections. But pigs actually don't have too many infections around. And remember that every year we give human patients unknown before infections from human donors. Rabies has been transferred in the last few years. West Nile fever has been transferred. All sorts of diseases that we give to the recipients, some of whom sadly die from them, because we didn't have time to check in the donor. What have they got? HIV has been transferred, hepatitis, and things like cytomegalovirus are transferred 90% of the time and can be damaging to the organ afterwards and life-threatening to the patient. Whereas with the pigs, who will be housed under perfect, ideal, isolated conditions, we will know that pig herd does not have hepatitis, they don't have this, they don't have that, they don't have the other. So we'll be putting in organs that we know are free of any infectious agent that might be detrimental to the patient. And then there are some less obvious reasons. When I was running a heart transplant program, I would see about 140 patients each year referred for transplant. Half of them I would decline to operate on, either because they weren't sick enough or because they had so many other problems that I thought they're not going to do very well. I don't want to use one of these very valuable donor organs on a patient who's probably not going to get through the first few weeks. So I would be very selective in the patients. Now, if we had an unlimited number of donors, we could be much less selective. We could say, well, maybe the patient's only going to live a year or two, but the quality of life may be good. We give them the opportunity of a heart. Whereas when you've got a very limited number of donors, you think, I really want a patient who's going to get through 10 years, not just 1 year. And so you're very selective. And in some countries, particularly Japan, for example, where they have all the technology and all the skills, there's a cultural barrier to deceased donation. And people just don't want to donate for cultural, religious, other views. And if xenotransplantation could take place in Japan, the number of transplants would go up perhaps 100-fold because they do so few. They've done less than 20 heart transplants ever in Japan. They do a lot more kidneys, but they get them from living donors, and similarly parts of the livers and so on. For example, heart transplant, say they've done less than 20 in 30, 40 years. And then when you look at people, say diabetic patients or patients with neuronal degenerative diseases, Alzheimer's and so on, Parkinson's disease, if you could treat those with cells, neuronal cells, you're talking of millions of patients. There's considered in this country between 1 and 2 million type 1 diabetics taking insulin every day, and another 20 million type 2 diabetics who have usually the diabetes that goes with obesity and older age and so on. So for those patients, we're never going to get enough donors from human sources, so we have to turn to something like the pig, and I'll come back to that in a minute. So we're interested in organs. We've done studies on liver, heart, kidney. My colleagues in Maryland work with closely doing studies on lung transplants from pig to human. We looked at islets for patients with diabetes. We're looking at corneas, even retinal cells for blindness, neuronal cells, I say, for Parkinson's, hepatocytes for certain conditions, liver cells, and even red blood cells. I believe one day all of the red blood cell transfusion will be from pigs, not from humans, because it would be much cheaper and easier. You won't have to do. They're now doing about 120 tests on each unit of blood that is going to be transfused, because to exclude all of these different infectious agents and so on. And in the pig that wouldn't be necessary, because you know they don't have the infections. So it will revolutionize medicine, to be honest, if we can get it to work. Now there's a whole number of barriers, of course, that's why it's not caught on yet. The immunological ones are the most important, but there are physiological, so safety ones about the risk of infection, ethical aspects of it, regulatory and so on. So let's just look briefly at the immunologic problems. If you do a pig organ transplant into a non-human primate, such as a baboon or a monkey, very, very similar to doing it into a human. By the way, pig hearts have been transplanted into humans in the past. One of my bosses in London did two one day when I was there. And pig liver's been transplanted into a human patient, and I think even pig kidneys have been transplanted. Here's a kidney from a pig immediately after it's reperfused with blood in a baboon. And you see it looks lovely, pink, red, well defined. Within 5 minutes, it will look like this. 5 minutes, it will look like this. It'll be completely destroyed, totally black, hemorrhagic, swollen, etc. And one of my former colleagues, Randall Morris, who was a cardiac surgeon himself, he said that when we're faced with this problem, this immunologic problem of rejection, he says there are 3 golden rules for achieving successful xenotransplantation. But then he added, unfortunately, we don't know any of them. And that was true at the time, but now we do know quite a few of them. But I've been working on it since the early 1980s, and I can see it. But so I think man is a very curious person, very good at research. And so we've got a good lot of curious people working on research in this field. That's why we now do know some of these problems that we face. And what happens with these pig organs, if this is the surface here of the pig, or the pig vascular endothelium, the lining of the blood vessels in the pig heart or kidney and so on, sticking out from its surface, it has some sugars. These are antigens, but they're sugars rather than they're carbohydrates. And we have antibodies against those sugars. And so as soon as you put the organ in, the antibodies bind to the sugars and activate a whole system, complement activation, all sorts of things, bring in cells, and it's destroyed rapidly. It's a defense mechanism because we've developed these antibodies when we, in our infancy, when our gut is colonized by various bacteria and viruses. You don't have these antibodies when you're born, but you get them within about 3 months. And it's a response to this colonization of bugs in the gut that we don't want them to injure us. So we develop all these antibodies against them to keep them in the gut. If they come into the blood, they're killed immediately. And unfortunately, the same sugar is on a lot of these bacteria as is on the pig organ. And that's why we have this dramatic response to it. So I say anti-pig antibodies are in primates, baboons as well as humans, are mainly directed towards a certain sugar. It's a galactose sugar, very similar to glucose, just a little bit different. And if you look at the human, we have 3 sugars on our blood vessels, the most important of which is the ABO blood group. We call it ABH, but the AB blood group. So if you are an A blood group and you get a B kidney put in, you will have anti-B antibodies and you will destroy that kidney very rapidly. There's about a 2/3 chance that you will destroy that kidney unless you've done all sorts of things to prevent that happening. And where we have the AB blood groups, or O, the pig has this galactose. So it's almost like a pig's blood group. But unfortunately, all pigs have the gal, and all of us have anti-gal. And so you get 100% destruction, not 60% as you do with the ABO incompatibility. And they have another one, sialic acid, which is identical. So this is the key thing, this gal against the ABO. That's why we destroy them. And you can see, here is the gal sugar on the right, and you can see how similar it is to the B sugar, except the B has this extra bit, and even to the A sugar. So it's very similar to the blood group incompatibilities that we're talking about. Now, we couldn't do anything about this, really, until we could genetically engineer the pig. And with these techniques, then, it was my own group when I was in Oklahoma City, actually, that we suggested in 1993 that maybe you could knock out the gene that makes this sugar. So then the pig will be just a pig, but it won't express this galactose. And therefore, there's no target for these antibodies. And it wasn't possible to do that then, when we first described it in 1993, but it became possible in 2003, and the company that I worked with were the first people to do this. And these pigs, fortunately, are very healthy, even though they don't express this gal sugar. And so this immediate rejection is now no longer a thing of the past. It is no longer present, it's a thing of the past, because there's no target now for these antibodies. There are other antibodies against other sugars, and we don't know what they are, but they're nothing like as important as this one against the gal. So this was a big step forward. So how can we overcome the other problem with the other sugars, because we don't know what those sugars are, so we can't knock those genes out until we know what they are. Well, when you bind the antibody, you set up this whole cascade of events that causes rejection. And one of the things are this complement system. And we do have pigs, though, in which we put in a human complement regulatory gene, so they're now protected against human complement. And so these pig organs now, I mean, for example, we don't destroy ourselves with our own complement system, because we have these regulators and protective genes. And so the pig doesn't have those against human complement. So we put into the pig one or more human complement regulatory genes. So we've now got pigs that don't express the main target, the antibodies, and when the other antibodies do bind, they're protected from causing injury because we've got these protective complement regulatory proteins there. So we have 2 ways of doing it, and luckily all these pigs are healthy, and they breed normally, and they seem just like normal pigs. They still have poor dining habits, but they go on in the same ways as any other pig. Now, obviously, when you put that organ in, even if you've overcome this immediate problem, just like when you put a human organ in, you can develop new cells and new antibodies against that organ, cellular rejection, antibody rejection. And so we have to have some good immunosuppressive therapy, just as patients with human organ transplants take, to protect them from these secondary rejection episodes. And the rejection of those is very similar to it as with a human transplant. And so we have these, what we call these gal knockout pigs, GTKO, and CD46 is one of these complement regulatory proteins. So where are we getting with this? Well, when we do transplants now, instead of them lasting 5 minutes, they're lasting 6 to 8 months. And there's no obvious rejection, either antibody-mediated or cellular rejection. But we are seeing some thrombosis occurring in the blood vessels in the organs, whether it's a kidney or a heart and so on. And we believe this is partly, there are some coagulation incompatibilities between the pig and the human. The pig's coagulation system is a little different from the human one, and it can't deal very well with human blood going through. When human blood goes through our organs, they don't thrombose up because it's all compatible. In the pig, its anticoagulants can't deal very well with the human coagulants, and so you get thrombosis occurring slowly over weeks or months. Here's an example. This is a pig heart. Normally, it should look a little bit like this, pink, nice blood vessels, but you can see here there's lots of areas around here where there's no myocardial fibers, and you can see there's clearly thrombosis in some of these blood vessels. And this is several months after the transplant, but it's destroyed by having multiple little heart attacks, really, multiple little myocardial infarctions. And that's the problem we're trying to sort out at the moment. And I say we can get out with the heart up to about 8 months, kidney up to about 3 months. The liver, for different reasons, we can only get to about 10 days, and the lung, for also different reasons, we can only get to 5 days. The lung is very fragile, and you get a little bit of thrombosis or whatever it is, and it's damaged very quickly. So we concentrate our attention largely on the heart and the kidney. Now, how can we solve this problem? Well, we're doing the same approach. We're putting into the pig human antithrombotic genes. There are genes that cause thrombosis, and there are genes that cause anticoagulation. So we're putting in some of these. I won't need to go into them. We've now got pigs, for the first time recently, we've got pigs with a thrombotic gene, a human antithrombotic gene. And to give you an example, normally when we put this, say, pig heart in, we will see that the fibrinogen, the platelet count here, the platelets drop down to a fairly low level because they're all being absorbed into the organ. They're all sticking in these little tiny clots that are occurring. And the first time we've done one with this anticoagulant gene in the red here, you can see that there's no drop in the platelets. So this is very encouraging. We're doing 2 more in October. So this is very encouraging that we seem to be overcoming some of these problems in the coagulation. And obviously we may need pigs with 2 or 3 or 4 anticoagulant genes to be optimal. And so we're working on that. Now, switching from organs, we'll come to pig islets. As mentioned, that this is the way to treat patients with diabetes. You give them new islets. The islets make insulin, and they make insulin when the patient needs it. They don't have to say, well, twice a day I'm going to give myself an injection. If you eat a big meal, your insulin will work because you've got the islets there which will produce the insulin. And I say there are between 1 and 2 million patients with type 1 diabetes, but only 7,000 or 8,000 deceased human donors. Your islets are in your pancreas. You've got 1 pancreas in each donor, so that means each year, at best, we could do about 7,000 or 8,000 transplants. And in fact, most of the patients who've had islet transplants need islets from 2 donors, 2 pancreases, in order to give them enough islets, because you lose some in the preparation. So we're talking of very small numbers. In fact, in the world, there's only been about 1,000 islet transplants done to date, and here we have a couple of million patients, even in the US. And the expenditure on diabetes is very similar to that even worse than that for kidney disease. We spend about 210 billion on patients with diabetes who've got complications of diabetes over their lifetime. It's at least 10% of expenditure. Now, pig insulin was used for many decades in patients. Before we could make synthetic human insulin, patients were treated with pig insulin. So we know pig insulin works. It's only 1 amino acid different from human insulin. So we know pig insulin works, so there's not going to be a problem there. It works in humans quite well. And so if we could put pig islets in and they could produce pig insulin, we know the patient would be fine. And now where are we with this? Well, let's just take our own experience. There are 2 or 3 other groups working on this with very similar results. If we take adult pig islet transplants into diabetic monkeys, if we use an ordinary pig, a wild-type pig, we correct the diabetes for about 6 weeks. If we use a pig with one genetic manipulation, we correct it for certainly more than 3. We've often euthanized the monkey because we couldn't afford to keep it much longer, but more than 12 months. And similarly, now we've got pigs with 3 to 5 genes, and we've got up to over 12 months. One's just been going for 15 months with no insulin at all. So we're getting close to a clinical trial. The trouble is we can't do it absolutely consistently. We get 2 that do very well, then we'll get 2 that don't do well at all. And we're not quite sure why it is. Probably something to do with the preparation of the islets, which are not easy to do. Here's the example of the most recent one. You see in blue is the blood sugar, and you can see on day 0 when you've got the islets, the blood sugar is now normal for the rest of the year. And the bars, the little bars at the bottom here, are measurements of pig C-peptide, which is an indicator of pig insulin. And we can differentiate between pig insulin and monkey insulin. And you can see it's continuing to put out a lot of pig insulin and C-peptide throughout the whole period of time. So we know it succeeds, and we know, and this was with fairly modest immunosuppressive therapy, and so no more than you give a patient with a human islet transplant. And here at the end of a year, these are islets in the middle of the liver. We put the islets into the liver for various reasons, and they're normal-looking islets, and there's no cellular infiltration, no rejection of them. So very successful. Now, what would we need to do before we could do a clinical trial of pig islet transplant? That's probably going to come before the organ transplants, because first of all, the need is greater, the risk to the patient is less, etc., etc. Well, we need clean pigs. We have to be sure these pigs were clean, did not have infections. So we need a special housing facility to stop insects coming in and all this sort of stuff, which would be expensive. Or maybe we could convince the FDA that if we had clean islets, if you take the islets out of the pig, the pancreas out of the pig, you isolate the islets, we culture them for a few days, and if you could show that those islets were free of any infection, it doesn't really matter what the pig had. So maybe we can get around with a less strict, if we could show that the islets that you're going to put into the patient are free of all infectious diseases. We need to have a clinically acceptable immunosuppressive regimen. It mustn't be too intensive, because the patient may get infections and so on. And finally, as I say, we need more consistent graft survival than we're getting at the minute. We're getting about 1 every 2, doesn't do as well as we like, and we don't fully know why. There is a trial in New Zealand at the moment, under the auspices of the Department of Health, going on where they're putting pig islets into patients with diabetes. But in this trial, they're encapsulating the islets in such a way that it protects the islets from the antibodies and the immune response. And so they don't have to give immunosuppressive therapy, which is a big advantage. I'm not sure that this is going to work, but people are working on this. And say in this particular trial, they haven't yet reported the results. My understanding is that they function for a few months, but then they run out of steam. The islets run out of steam, probably because they're not getting enough nutrients, or because the capsules are not good enough at protecting them. But it's another approach. So we are already doing clinical trials in patients of pig islet transplantation. Now, there are all these other things, physiological, how these islets how these organs are going to function and so on, the safety of the risk of infection and so on and so on. And I'm not going to go into many of those. I'm just going to give you one example of some woolly thinking, how people don't think clearly. There was a group in Britain that was asked some years ago, how many human genes can you introduce into a pig before the pig is no longer a pig? I mean, if you're going to put in, you don't want to change the pig too much. So they, led by a leading academic lawyer who has been knighted, he's now Sir somebody or other, because of his contributions to law, they come up with this. They said, there are limits to the extent to which an animal should be genetically modified. And I think we probably all agree with that. There are limits. You can't overdo it. This was 1996. And then they added, pigs should not be genetically manipulated beyond a point that we would regard as intolerable to the animal. A moral point of view concerned with the pigness of pigs, which comes a point where the pig is no longer a pig. Now, if you understand what he's talking about, please come and tell me afterwards. I have not a clue what they're talking about here. And I think they think they've got any idea what they're talking about. This pigness of pigs is ridiculous. So there was a similar group in the Netherlands, and they came up with a much more sensible idea. They said there should be no unacceptable implications for the health and welfare of the animal. You don't want to manipulate the pig so that he's not healthy and doesn't feel well. which luckily we haven't done. And there should be no serious ethical objections to the procedure. For example, you shouldn't manipulate the pig's brain to become more like a human, or the human brain to become more like a pig, and there should be no genetic changes that might lead to the reproduction of one species by the other. So I think these are things we'd all agree on. But the British approach was not quite as clear-cut as this. Now, although we're getting a long way, I think the pig has probably got a few more tricks up its sleeve that we'd have to overcome. They say it's like peeling an onion. Every time you take off a layer, you find there's another one underneath, and it's always accompanied by tears. And so it's probably like that. But I like this saying by the former chairman of Rolls-Royce company in Britain: there are many ways of losing money. Women are the most fun, gambling is the fastest, research is the most certain. So we need more money for research, but not all of it will be well spent probably. This is Sir Roy Khan, who was one of my mentors when I was a very junior surgeon. He's still alive. Last week or the week before, he won the Lasker Award, very prestigious award, with Tom Starzl for their contributions to development of liver transplantation. I worked with him in the very early days of liver transplantation in Britain. He says clinical xenotransplantation patients is just around the corner, but it may be a very long corner. And as he said this in 1995, he got it right, because here we are nearly more than 15 years later, still trying to solve all the problems. He once told me, if you're doing research in xenotransplantation, you've got a job for life, which was not that encouraging, although it was supposed to be. So this is what we've been like for the last 30 years, holding back these waves of immunological barriers and safety problems and so on. But we are making some progress. And I like this quote. It's a little like wrestling a gorilla. But you don't quit when you're tired, you quit when the gorilla is tired. And that's what we're trying to do now. We're waiting for the immune system to run itself down, and then we will have won. Now, I worked in Oklahoma City for a while. This was some years ago, and I tried to do the best I could to be a good Okie and wore some cowboy boots and a cowboy hat. And they have a very nice, they have a lot of Native Americans there who are very bright, and they have a very nice quote here: timing has a lot to do with the success of a rain dance. There's no point in doing a rain dance unless you looked at the Weather Channel to tell yourself it's going to rain. And they're very insightful. And I think with xenotransplantation, I think now the timing is getting right, because we now have all this technology to manipulate the pigs. And we didn't have this even 5, 10 years ago. And so I think we're much more likely to get success now than we did 10 years ago. In fact, we are beginning to get success. So I don't think it's going to take too long now, because the technology is improving every month or two. So to quote Dan Quayle or Yogi Berra again, I think the future will be better tomorrow. Again, whether it's the future tomorrow or the tomorrow of the future, we don't know. Now, there are a lot of similarities between humans and animals, so I think it's ordained that we should actually be able to get this success. When you're little, you tend to mimic animals. You argue over certain things. You mimic cows, they mimic us. When you're at school, you always have a big fat friend, and that's the animal world as well. Sexual harassment seems to occur in the animal world as well as in the human world. Crime doesn't pay, whether you're an animal or not. And in England, where I come from, everybody has a dog, and you will eventually begin to look like your dog anyway. So. And some years ago, Time magazine said, well, men are pigs anyway, so really what are we arguing about? And I think there's probably some truth in that. So one day, making a pig of yourself could take on a whole new meaning, and I'm hoping it will do. And Willem Kolff, who designed the first artificial kidney, the first dialysis machine, which he did in Nazi-controlled the Netherlands during the Second World War, which was quite something to do at that time. He died just a year or two back. He said that even without hope you should undertake, even without success you should persevere. And so that's what we've all been doing for these last few years. So I'll leave you with this last quote, which is from George Orwell's Animal Farm novel, considered the greatest novel of the 20th century, at least in Britain it was. And he said that creatures outside look from pig to man, and from man to pig, and from pig to man again. But already it was impossible to say which was which. And I think one day a patient will walk into the doctor's office and with a kidney transplant, and the doctor will not know whether this is a pig kidney or a human kidney, because it'll be working just the same. Thank you.
Wow. Unique opportunity. What questions might you have? Not going to surprise me if nobody has one.
So with our immune system and stuff, what I'm always sort of the problem is that there would be something in the pigs that our immune systems would not be able to fight off, and we'd have something that would kind of jump species.
We have
you know what
something
I'm talking about?
Jump species.
No, I don't see that anything in the pig that we can't change to make it more like the human. You know, the technology is getting there. For example, now they can put the gene just in the islets or just in the endothelium of the blood vessels, not in the whole pig. So there are some genes that if you change the whole pig, the pig doesn't survive. It dies in fetal life. But if you put it just in the islets or just in the brain or just somewhere else, then it's fine. And yet, when you take those islets out, that's where you want this gene, and you put them into the recipient, and they're protected. So they're getting the technology very clever. So I don't think there'll be a thing that we can't do if we, in due course. Was there another question?
Yeah.
Yeah.
You said that the pork insulin was used because of the similarity between the three. Now, they also use beef insulin.
They also use beef?
Beef, I believe, is one of the types.
For this purpose?
Just for insulin. So they.
Oh yes, they could.
So
yes.
Is there a similarity between some sources of beef?
Yeah, insulin itself is very similar throughout most of the mammal world.
Okay.
And so you could use beef insulin. We're not using cattle for this thing because pigs are much easier to manage. I mean, you can keep them indoors under clean conditions. To keep cattle are much larger, etc. It would be difficult. And so pigs, when they start, they're very small, so you can take a heart, put it in an infant. They get to 1,000 pounds, a big pig. So you can get a big organ if you wanted to. Yes.
I was wondering if you had any comment on the construction of organs through like stem cells, and whether that might actually be this method.
The competition for xenotransplantation is either mechanical devices, which have got quite good in helping the heart, but I still believe that a normal biological heart is going to be better in the long run than a mechanical device. I think with thrombosis occurring and so on in these mechanical devices. Or stem cells, and the idea is that you make an organ from the stem cell. Now I think you may be able to make islets. In fact, people are trying to do that. It takes a number of steps to do this, but you could make islets, and you could make them from a stem cell from the patient who's going to do them, so they won't be rejected. One of the problems there is, though, that most juvenile diabetes is due to an autoimmune disease. So if you put in the same islets, they might be susceptible to the disease, whereas we think pig islets won't be. So there's a difference there. But I think to get a whole organ from a stem cell is going to take a long, long time, at least 50 plus years. I mean, you think how complex a kidney or the liver is. And I think to actually think you're going to create a new organ that's going to take the place of a human organ is going to be very difficult. So I think that xenotransplantation may come in first, maybe in 50 years' time it'll be superseded by something better. Gentlemen,
why the pig?
Number of logistic reasons. I mentioned the size, their breeding capacity. They breed in 10s or 12s, whereas cattle breed in 1s or 2s. So you get a large number quite quickly once you've got the sort of pig you want. They're easy to house, etc., etc. So there's really logistic reasons that we've selected a pig. We probably could have chosen some other animals like the goat or something, but pigs been. And the technology in the pig is much more developed than it is in any other species at the moment. Yes, you.
Henry.
Yeah. If you develop a pig that has the right makeup and they breed, whatever the makeup is will be in the
it's transferred
offspring.
in the germ line. And what we do, of course, we've got one pig with 4 genes in another pig. This is what we're planning on doing. Another pig with another 4 genes. We breed them together. We end up eventually with pigs with 8 genes, which will speed up the process. But the the moment we can put a couple of genes in at one time. We're working on putting 4 in. We can't yet quite do that, but if we could do 4 at a time, and we've got 2 pigs with 4 different genes in, and then you breed them together, and you would end up with pigs with 8 genes. So yes, they're not lost through after pregnancy. You won't have to clone every animal who's going to be a donor. You just breed them.
Don't you expect to have a lot of problems from animal rights?
I suspect we will have problems with animal rights. I would think the public would believe in general that the benefit of having this available to patients with terminal organ disease outweighs the animal rights groups.
Well, you and I would think.
Yes, but for example, if they don't want pigs to be used, remember these pigs will be housed under perfect conditions, ideal conditions, probably as good as any animal ever been housed, except maybe very expensive racehorses. They will be slaughtered, not in the slaughterhouse where they're hit in the head with a ball and their throats cut, but under an anesthetic. So there'll be no pain. And if we're killing 100 million pigs a year for food, can they really object to a few thousand a year for organs to save people's lives? So I think they won't get a lot of support.
Well, I hope you're right, because in 1992, when I had my liver transplant here, and there was research going on, outside my window, I was telling Jim this earlier, there were people protesting all the research on xenotransplantation.
But what they were objecting to mainly there was the use of baboons, because Dr. Starzer was using baboon as the source of liver transplants. And I think that the attitude to pigs is a little bit different from non-human primates.
Right. Hopefully.
So I think this British committee that I made fun of, they said they didn't think we should use non-human primates for this purpose, but they said they thought it would be okay to use pigs. Yes, sir.
One more question. Is there adequate funding supplied for this research from government sources?
There's never adequate funding, of course, as that said. We're reasonably lucky. The National Institutes of Health has a grant specifically for xenotransplantation. They felt it was important enough to do this. And we are one of 3 groups that have funding from that. And it's on a 5-yearly basis, and luckily we got it renewed a couple of years ago. But of course we're never sure if the economic situation drops. They may decide they don't want to fund this specifically. And so we may run out of funding. So we certainly are looking for funding because I'm sure we would have solved all the problems by now if we'd had really good funding over the last few years. Because we've got the technology, we know what we want to put in or to take out, and we just haven't had the people at the time and the money to do it. So there is certainly a big need for funding, both in this small company where they make the pigs and with people like us who are testing the pigs and suggesting what the next step should be. So there's never enough funding. And I'm actually a little, in immunology, in transplant immunology, to some extent, and I don't want this to get further in this room, but the reviewers of these grants often are immunologists who are very much mouse doctors who looking at minutiae about different cell types and so on. And I sometimes think they don't see the big picture. They don't see this will cure patients. They're more interested in does this cell do that or does it not do that? Sometimes it doesn't matter whether it does it or not. You know, it's not going to affect any patient care. And I think there's some of the funding. I put in grants, not recently, but some years ago, which I thought were good applications for funding to some of this work. And the reviewer didn't have any scientific criticism. And I thought, oh, that's good. They think it's good. And the last line said, but is xenotransplantation ever going to work anyway? So we're not going to fund you. You know, so there's a mindset with even some scientists that just don't see the big picture. Yes.
Don't they already use pig valves in some cases?
Pig valves have been used for many years. That's another thing about the animal rights people. Haven't complained about that, and yet that requires slaughtering the pig. They process those valves, so they generate a relatively low grade of rejection because they're processed in chemicals. I've put many of them in patients. But they do fail, And in young people who have a good immune response, a very active immune system, they fail sometimes within 2 or 3 years. If you get to my age, they will take 10 to 15 to 20 years to fail because the immune system is not so active. And this was a big problem, particularly when I was in Cape Town, because a lot of our black patients had rheumatic heart disease, they needed a valve replacement in their teens, and we transplant them at the age of 13, they'd be back a year later with wanting a new valve because the valve was already rejected. Some of these new pigs would actually solve that problem. But, and I've contacted some of these companies that make these pig valves for pig valve surgery, and I've said, look, we have much better pigs now. These pig valves would last a lot longer. And they say, well, how much are they going to cost us? And I say, well, they're probably going to cost you a few $1,000s because these pigs are, very expensive to develop and so on. And they said, the pigs valves we get from the slaughterhouse, we pay $1 each for each pig valve. So they said, we're not interested in paying, thousands. And if you say, well, they're going to last longer, the company says, we don't really want them to last longer. We want them to put them in again. So from a commercial point of view, it's not going to benefit them at all. So there's that resistance from the commercial side of it. It's not going to make a big profit, and they may last longer, which is not going to be in their best interest. So there's a number of factors. Yes.
So this is kind of just a personal question, but do you go in the lab every day with a white lab coat, or are you more in the...
I never wear a white lab coat, but I do go to the lab every day, and I do go to the operating room with my junior colleagues when they're doing a heart transplant or a kidney transplant in the pig to the monkey model. I'm at the stage of life that I direct everything, but don't do anything. Keith Rinsma, who I mentioned, he said, you get to the stage of life that you pick the best people you can around you, you give them the facilities to do it all, and when they do some good work, you take all the slides and you give a lecture somewhere about them. And that's what I do now. But, so I'm very much involved, but I'm not doing any much hands-on stuff myself.
You write books.
I write grant applications, take a lot of time, and we write a lot of papers. Many of my research fellows are from abroad, like Japan or something, China. Their English isn't too good, so I find I have a big role to play in presenting their data as they would like it presented. Yeah.
David, I can't thank you enough.
Pleasure.
And I knew I had that article. Here, step over here.
Yeah.
Professional photographer Susan
okay.
is catching us. And
you're going to disappoint a lot of people. You keep referring to me, though.
He's very humble.
Go ahead.
I'm good with the camera. I knew I had that article, and I put it on the next
in
slide.
The Scientist. It's in the Scientist.
Right, and so here's the article for people to take a look at it. Like, I knew I had it someplace to post. Right? And that's how God worked with us to bring this powerful presentation to us. I mean, somebody said, Jim, you've done such a great job getting these presenters. Believe me, some of them fall into my lap, such as you. That was fantastic. And on behalf of TRIO, I'm going to give you this book. Great quotes from great leaders.
Oh, excellent.
And in the back is a DVD. You can put it in your computer and enjoy that.
Thank you.
And start quoting some.
Well, as you saw, I like quotes.
You are a quote person.
I like one thing. We're in this core facility, donor facility. When this comes about, this place will close down. There'll be no need for any more. I always tell the organ people you're going to be out of a job soon as
They don't seem to be too worried about that.
Thank you very much.
My pleasure, Dave. Thank you.